Hi, I'm the CEO and Managing Director of Arovella Therapeutics. We're an Australian Stock Exchange listed biotechnology company under the ticker ALA. And I'm delighted to be presenting today on our company, where things are at currently and how we see them shaping up moving forward. I'd just like to say a special thanks to the organisers of the JP Morgan BIOTECH Showcase. And of course, a thank you to everybody that's taken the time to dial in to listen to our company presentation today. Thank you. There's the usual disclaimer. And just introducing Arovella Therapeutics, as I said, we are a biotechnology company, and our reason for being is that we are committed to helping people live longer and healthier lives. And the way we're intending to do that is by working with therapeutics that really we see are at the cutting edge or the forefront of a particular area, in our case, oncology. And so what we're working on currently is realizing the potential of invariant natural killer T cell therapies. And just to point out very clearly, invariant natural killer T cells are a naturally occurring subset of our immune system and our immune cells, and they are functionally and phenotypically distinct from both T cells and natural killer cells. But I'm looking forward to taking you through the platform. Now, in terms of our company, we do have a number of key highlights that I would like to point out. As I said, we're working with an CAR-NKT cell therapy platform. We licensed that in the middle of 2021 from Imperial College London, and that's a top 10 university worldwide. And we also, at the end of 2021, licensed in a novel monoclonal antibody targeting a DKK1 peptide that can be used as a CAR, and that was licensed from MD Anderson, one of the world's best cancer centers. So now we have an allogeneic platform due to our iNKT cell therapy with two targets, the first being CD19 expressing lymphomas, and the second target being for cancers that produce DKK1. As a business, we're going to leverage the experience we have in our senior group to source, evaluate, and acquire innovative technologies that we believe align with our focus areas. We're very much a data-driven organization, and we think this is fundamental because good science underpins good data, and that they both underpin the development of outstanding therapeutics. So we use data as a key driver for making decisions about the key assets that we'd like to bring into our company, but also when thinking about structuring clinical trials and so forth. We have a world-class team with a board and senior management group that has extensive experience in drug development, and with some of the recent hires that we brought into the business, particularly with cell therapies. Arovella is the only ASX listed company working with an iNKT cell therapy platform, and one of a handful worldwide. And as far as we know, we are the only company worldwide with a chimeric antigen receptor targeting the DKK1 peptide. So as I mentioned, I would like to talk more about our iNKT cell therapy platform and what it means and what it is exactly. Before I do that, I'd just like to give a brief overview of cell therapies to set the scene. So this is something I'm sure most people in the audience will be familiar with. There are now 5 approved or FDA approved cell therapies. So it's been a big paradigm shift in the way we think about cancer treatment, and so exciting because of the pronounced cure rates that these particular therapies are having. And when we typically think about cell therapy, we often think about the fact that the cells need to be taken from the patient and given back to that patient. Well, that's where there's a fundamental difference for the iNKT cell therapy platform. We expect the starting material to come from a healthy donor, which may be collected at a hospital or clinic, and then the steps are quite similar in that we select and isolate the iNKT cells. They're then genetically reprogrammed to produce a chimeric antigen receptor, or CAR, on their surface. They're then grown up into sufficient numbers. Again, there's a change here for us in that we would freeze down those cells ready to be administered to a patient. When a patient is deemed eligible for the therapy, they're given to that patient, where then they're able to go out and find the target that we've given them the CAR to recognize, and then they trigger the destruction of those cancer cells. And so when we think about the starting material being a blood sample, we do know that there are a whole range of immune cells within that mixture. So we've got macrophages, natural killer cells, T cells, and of course iNKT cells, which is what we're focusing on. And a good way to think about this is that they are really the soldiers of the immune system, and their job is to go around and find things that don't belong, whether that's an infectious disease or in our case if it's cancer, and eradicate that material. And for us, we've depicted deliberately here the iNKT cell as an armed soldier. That's because they are one of the most potent naturally occurring immune cells. And they bridge the innate and the adaptive immune system, but importantly they're already equipped to naturally target and kill cancer cells, and that's by virtue of the invariant T cell receptor, or ITCR in short. And as I said, that targets something called CD1D that's found on numerous cancer types and enables them to trigger the destruction of those cancer cells. And not only once they activate it, do they actually help to eliminate cancer cells, they also activate other components of the immune system like T cells and natural killer cells, which has an added advantage and enhances the anti-cancer effect. Now the ITCR is also something that doesn't change between individuals. It's what we call monomorphic. And that actually means that iNKT cells can be given from a healthy donor to a patient, because they not only do they not cause graft versus host disease, they suppress it, which is again unique to this cell type. And they show significantly improved killing of cancers that produce CD1D when we give them a chimeric antigen receptor, and it is far more effective relative to conventional T cells, and I'll show you the data for that in just a moment. And so when we look at the comparison between CAR T cells and CAR-NK cells, we see that there are numerous points that stand out for the iNKT cells. So they are a subpopulation of T cells with natural killer cell properties, so that's quite unique. They already have an intrinsic anti-cancer T cell receptor, so when we put in a CAR, they are dual targeting. Persistence will be assessing as we move and develop these products into clinic. They don't require extensive genetic engineering to be used off the shelf. They are the only immune cell that naturally suppresses graft versus host disease. Early clinical data from others suggests there is low risk of cytokine release syndrome or neurotoxicity. And as I mentioned earlier, we're developing this to be used off the shelf, which we think will improve logistics of the therapy, but also have a better starting product because we're not working with immune cells that have been subjected to cancer or chemo or radio or other forms of anti-cancer treatment. And so just to go through what it might look like on a molecular level with a cartoon. So as I mentioned, we start with our iNKT cell therapy cell. It already has the ITCR, which can recognize CD1D. We then equip the cells with a chimeric antigen receptor, and this will be used to target a particular cancer of interest. They are then frozen down, ready for use. When the cells are then given to a cancer patient, the ITCR will find CD1D that's bound to a glycolipid, and the chimeric antigen receptor will find its target on the cancer cell. And once this happens, we have the release of components like granzyme B and perforin, which then trigger the destruction of the cancer cell. And of course, what's not depicted here but also takes place is the release of cytokines and other signaling molecules that bring other immune cell components into the fight against the cancer. And as I said, this is a lovely model of how this works, but I'd just like to show you the data that demonstrates that this is actually the case, that we do get better activity. This data was published in the prestigious journal Cancer Cell. And so this product it's a CD19 targeting cell therapy, iNKT cell therapy called ALA-101, and we are anticipating this to be an effective off-the-shelf cell therapy for the treatment of CD19 expressing cancers. Now there's a fair amount of data on this slide. I don't want to go through it all in great detail, but just to highlight the three important points. The first is that when we look at a range of different therapies, we see that CAR-19 iNKT cells have faster killing of cancers that produce CD19 and CD1D. And what we're looking at the bottom panel, anywhere there's fluorescence, that means after 3 days of treatment, the cancer is still present. And when we look at the last column, we see that for the iNKT cells, essentially after 3 days of treatment, there's no fluorescence, which indicates that tumours have been cleared. So rapid clearance is point 1. Point 2 is equally as important. Rapid clearance is great, but we need that to translate into long-term survival. So what we see here, this is a survival curve looking at the different treatments, and what we're looking at is survival over time, and we see that approximately 95% of the CAR-19 iNKT cell treated mice are alive versus 60% of the CAR-19 T cell, so the conventional cell therapies. So it's approximately 1.5 times more animals surviving. And then the third point is what we call a spontaneous secondary remission. In 4 of the animals treated with our therapy, the cancer started to return to the brain between 20 to 30 days. Remarkably, with no further treatment at all, the cells were reactivated and the cancer was cleared a second time spontaneously. So we believe that this really validates, or this data really validates the use of the iNKT cells as a treatment, not just for CD19 expressing cancers, but others that also we can find CARs to target and also produce CD1D. So for that program, we're expecting to commence manufacturing of the plasmid and the lentivirus in this quarter, and certainly looking forward to developing that towards clinical trials. And I'd like to talk about the second half of the equation, which is the safety of the therapy that's been gathered to date. So this is some of the preclinical safety evaluation work that was completed. Essentially, after giving the mice the CAR-INKT cells, there was no body weight loss, nothing to indicate off-target effects, no histological changes in normal tissues, and really besides the profound anti-lymphoma effect, there wasn't really any evidence of negative pathology findings, again indicating that the therapy is likely to be quite safe. And just taking that a step further, as I mentioned earlier, conventional CAR T cell therapies are limited to autologous, so they must be given from the same patient back to that patient due to the potential of the potentially fatal graft versus host disease. The only way around that at the moment for T cell products is through genetic engineering, which we don't have all the answers yet in terms of side effects or even the impact on the fitness of the immune cells. But this is literature not just from our group, but from many different groups demonstrating that not only do invariant natural killer T cells not just cause graft versus host disease, they actually intrinsically protect against it. So that is quite unique to this cell type. And what it means for us is that our iNKT cell therapies will be developed as allogeneic, and they'll be done so requiring only the minimum level of genetic engineering, which is designed to produce the chimeric antigen receptor. And so looking at use of iNKT cells in clinical trials, so they have been used for more than 15 years in the presence and absence of chimeric antigen receptors in a range of tumor types, including blood cancers and solid tumors. To date, the efficacy data is encouraging in both forms of cancer, and really importantly, the side effect profile is also equally encouraging, where we've seen low risks of cytokine release syndrome and neurotoxicity, and no evidence of graft versus host disease for allogeneic iNKT cell therapy products. And I'd just like to switch gears now and introduce our second major cell therapy product, which is ALA-104. So to give you some background, it's a monoclonal antibody that targets a fragment or a peptide of a protein called DKK1. So DKK1 is a secreted protein that functions as a negative regulator of the Wnt signaling pathway. And what's been shown is that DKK1 is overproduced in numerous cancers, and in our case, we're not targeting the secreted protein. That would not work as a cell therapy because it wouldn't be stuck on the surface of the cancer. But what we're actually targeting is DKK1 peptides. And so as I said, what happens is the cancer produces DKK1. That gets chopped up into small fragments by something called the proteasome. And I know this is rather crudely drawn, but the peptides get loaded onto a HLA-A2 complex before they get shuttled. And then the HLA-A2 complex with the DKK1 peptide gets presented to the surface. And it's this target here that our monoclonal antibody specifically recognizes. And of interest, 40 to 50% of the population is HLA-A2 positive, so that's already quite a large market size. A number of different cancers produce DKK1, so we believe that there's quite a large opportunity across a number of different cancers for this product. Now, taking this a step further and looking at DKK1's role in cancer, as I mentioned, it is overproduced in a number of cancer types, and this is just naming a few. There are others, but this includes pancreatic, esophageal, hepatocellular, breast, lung, and multiple myeloma. And in addition to that overexpression, it's been shown that DKK1 can indicate poor overall survival as well as shorter disease-free survival. So DKK1 has recently emerged as a potential biomarker of cancer progression and prognosis for a number of different malignancies. And now there's also growing evidence that DKK1 plays an essential role in cancer progression. So we are quite excited about the target, given the breadth of cancers and the role that it seems to be playing in this disease. And so where we are today, the DKK1 monoclonal antibody was developed at MD Anderson, and it's been shown that it can be incorporated into a chimeric antigen receptor. Just to step back though, the DKK1 peptide targeting monoclonal antibody itself has demonstrated activity against cancers like multiple myeloma and breast cancer. But what we've been able to do is take the short chain variable fragment of that monoclonal antibody, incorporate it into CAR T cells or a chimeric antigen receptor into T cells, and it displays excellent activity against blood cancers such as solid tumors. And so that's what's been depicted here and is already complete. This is a schematic of a T cell that's got a DKK1 CAR, and the animal models demonstrate activity in multiple myeloma, pancreatic cancer, lung cancer, and breast cancer. So what we're going to be doing moving forward is taking that CAR and putting it into our iNKT cell therapy platform. And initially we're going to be targeting multiple myeloma, and I'll talk why we're doing that in just a moment. But there is the potential also to take that into solid tumors. So as I said, data underpins our decisions, and this is a snapshot of the data that we've seen to date for the DKK1 CAR T cells. So this is looking at a very specific blood cancer, multiple myeloma, and using 3 different animal models for this particular cancer type. And what we're looking at here are 3 survival curves, where on the vertical axis we have the number of surviving animals, and on the horizontal axis we have the number of days they've survived. And in all models, the black line demonstrates the animals that were not treated. We see that they've all succumbed between 30 to 40 days, but the red line, which represents the DKK1 CAR T treated animals, they're all alive between 50 and 60 days. Now, why we're quite excited by this is that multiple myeloma is a cancer that also produces or expresses CD1D. That's the target of iNKT cells. So we actually believe that by incorporating DKK1 CAR into iNKT cells, this will make them dual targeting and will have a better performing product. And also, I mentioned it earlier, and I just haven't shown the data for it, but there's good data to support that the DKK1 CAR T cells have activity in lung, pancreatic, and triple negative cancer models as well. Now, in terms of preclinical safety, Professor King Yi, who's now at Houston Methodist, has demonstrated that the CAR T cells only kill those that have the DKK1 peptide presented in an HLA-A2 complex on their surface. They do not kill healthy blood cells, and when used in animal models, they are considered safe, and it doesn't look like there's any off-target effects. In terms of the specificity of the monoclonal antibody, it's been tested against a panel of 35 normal tissues, and only one of those tissues was non-specifically targeted, which was the tonsil. Moving forward, Arovella will confirm that the DKK1 technology does not target healthy cells, but as I said earlier, we'll rapidly look to combine this with our iNKT cell therapy platform. So in terms of IP, it's quite a long life patent out until 2039. It's been filed in all major territories such as the US, Canada, Europe, China, and Australia, and we've already received a favourable search report. So in terms of development, over the next 24 months. It is going to be quite a busy time. In blue, I've just depicted the CAR-19 iNKT cell milestones, and in pink, it's the DKK1 CAR-INKT milestones. So key next steps for the CAR-19 iNKT cells are to select our GMP manufacturer and to complete GMP manufacturing of CAR-19 iNKT cells for phase 1 clinical trial, to get FDA clearance, IND clearance to initiate a clinical trial and to dose the first patient for CD19 producing cancers. And for DKK1, key steps are to confirm safety and specificity of the CAR, as well as combine it with the iNKT cell therapy platform and demonstrate activity of those cells in models for cancer such as multiple myeloma, as well as potentially solid tumors, and then define the manufacturing strategy for the DKK1 CAR-INKT cells. And something that we'll be always continuing to do is to look to acquire or license additional CARs or technologies that we believe are complementary to the iNKT cell therapy platform. So I'd like to highlight some of the iNKT cell players and the commercial activity that's transpired only quite recently. And just a bit of context, when we think about T cell or NK cell players in this space, since we had the first approvals of these products in 2017, there are now hundreds of those companies, which is exciting for the field, but does make it quite competitive. Whereas when we look at the iNKT cell players, there are 4 companies that are targeting cancer indications. So we have Appear Bio, who are Preclinical, and they raised $52 million in their Series A in May of 2021, and shortly after that announced a strategic partnership with Kite Pharma, and that deal had a value of $875 million. Cure Therapeutics, who are still early stage phase 1, they were acquired by Athenex in May of 2021 for $70 million upfront and $115 million in milestone payments. Mink Therapeutics, also in phase 1 for non-CAR-INKT cell therapy products. They raised $40 million in October last year and now have a valuation of about $150 million. And Arovella, as I said, we acquired the license to the platform Imperial College in June 2021, and then the DKK1 targeting or peptide targeting monoclonal antibody from MD Anderson Cancer Center in December 2021. And we have a valuation of about $19 million. So we do see that there's a fair amount of room for growth for Arovella. And I'd just like to finish by going through our key leadership group and our pipeline. So we have a board that's got a lot of experience in drug development, right the way through from taking early stage development products and all the way through to commercialization. So our chairman, Paul Hopper, he has certainly been involved with numerous early stage biotech companies from development and been involved in trade sales to companies like Merck. Myself, I have a background, I have a PhD in biochemistry before a really fairly lengthy research career before moving into drug development and then into venture investing before joining Arovella Therapeutics. I'd like to highlight the 2 new directors that have joined the board in the last few months, Dr. Deborah Barton and Dr. Liz Stoner. Both are phenomenal individuals, and we couldn't be happier to have them on the board. Dr. Barton is a medical doctor by background, where she specialized in oncology. After doing that for some time, she moved into senior clinical roles at companies like Celgene and Novartis. And more recently, Dr. Barton has been involved in cell therapy companies like Iovance, and she's now currently the Chief Medical Officer of Charisma Therapeutics. And Dr. Liz Stoner has a research background, having formerly been Assistant Professor of Pediatrics at Cornell University, before moving into, again, clinical and operational roles at companies like Merck. She's currently an executive partner at MPM Capital. Again, it's lovely to have somebody that's gone right the way through from that early academic research to the commercialization of technologies. And Liz was the interim CEO of a company called Semma Therapeutics that went on to be acquired by Vertex for $950 million. And so we've got David Simmons, who is also a former senior audit partner with EY, and David Phillips, who has a large amount of experience, more than 35 years in the healthcare sector, spanning large pharma, biotech, but also venture investing during his time as managing partner of SR1. And so in terms of our pipeline, we are quite excited. In the last 6 to 12 months, we've really built out an exciting cell therapy platform where we now have two leading products that we're developing, ALA-101, which we're developing to target CD19 expressing lymphomas, and ALA-104, which we're also developing to target particular malignancies that express DKK1. So both of these are currently in the preclinical stages, and we're aiming to develop those into clinical trials. But as I said earlier, we are also excited that we'll continue to look to expand on those particular products and our pipeline. Now, I haven't talked about it today, but we also have a second platform, our Oromist platform, which we use to take drugs and convert them from solid dose form into oral sprays. And our most advanced product in that space is Zolpimist, which is for short-term insomnia, and we're expecting that to be on the shelves in calendar year 2022, which is exciting. And so with that, I'd just like to say thank you very much again to the organisers of the JP Morgan BIOTECH Showcase. A big thank you to everybody that's taken the time to listen in today. If there's anyone that would like any additional information, I would encourage you to go to our website, arovella.com, and certainly be happy to provide any additional information. Thank you.